Okay, are there any questions before we begin? A bit about the grading. I now have lab 4 graded for everyone. So please work on that. And it's due this Saturday at 11:59 p.m. for the second grading. I have a few other things graded. Let me see if I can figure out what it was. The student discussions are graded. I now open them up so you guys can use it again to discuss among yourselves. I will not generally answer questions in the student discussion. If I do go in, the only time I'll answer something is if somebody has supplied a wrong answer. But you may use that for your own discussions. I have given extra credit point for those who either asked a question initially or answered a question. There are a few people who got 0 because you neither asked a question or answered a question. And then some people who didn't obviously respond, they obviously got nothing also. But and now if you put something in the student discussions, you won't get extra credit, but you certainly are free to use the student discussion. I've now opened it up again. I have extra credit study 2 graded and extra credit study 4 graded. I need to grade extra study credit 3. And I have lab 00, lab 01, worksheet for lab 2, worksheet for lab 3, worksheet for lab 4, graded the first time. None of the labs have been graded the second time. And I have not graded worksheet for lab 5. So I believe I've already mentioned that I'm postponing lab 5. The second grading of it will not be due until the 17th, and that's assuming I get the first grading done before Tuesday. The lab 4 was mostly graded on Tuesday, but there were a few students who were graded on Wednesday, and I apologize if you were one of the late students, but lab 4 is now completely graded. Any questions about the grading? The practice quiz 00 and 00A and 00B. I do need to get that graded. I'll try and work on that tomorrow. All right, any questions about the grading? I do make myself available in the lab to answer questions, give people direction. And so if you're struggling in the lab, you should take advantage of me. That's why I stay there in the lab until the last student leaves. So far, I've never had a lab where I ever logged out and there are still students there. So I do make myself available. Use me if you're struggling in the lab. A number of you are, and you should try and use me because you're not doing too well in the lab. Now, obviously, you will get the lab graded a second time, and hopefully your grade will improve. All right, shutting down the grade book, and now I will share my screen. Let me see, I've got some emails here. Somebody asked about the lecture and the lab were not available from last Tuesday. My apologies there. If ever I don't have it placed on YouTube a day later, send me a note as a reminder to get it up. I went there to put up today's lab and lecture actually today's lab, sorry, and yesterday's lab and lecture, because I knew I didn't get those up. And I was surprised I hadn't gotten anything up for Tuesday. So I now have the Tuesday class up. I don't have the Monday, Wednesday, or the 3 p.m. lab up yet. All right, let me share my screen. We will begin. Can somebody confirm you're hearing me? I guess I should have said that in the beginning. Why is that not sharing?
Yes.
Thank you. All right, today is February 8th, and we should be working on chapter 5. Note that next week you will have a quiz 3, on Wednesday. I'll leave that up for the lab later today, but let's go to chapter 5. I have the last slide we worked on was right here, where I showed you that how enzymes reduce the activation energy when there's an enzyme around. It's much easier to get the reaction to proceed than when there's no enzyme around. The activation energy is much greater. So let's talk a little bit about enzymes. Enzymes are biological catalysts. A catalyst is something that speeds up a chemical reaction. You don't need to know how many times, but up to 10 to the 8th or 10 to the 10 fold. Enzymes are encoded by genes. So they are things our cells make which speed up biochemical reactions. They are specific for a single chemical reaction. That's a little bit of an oversimplification. Those of you who know about drugs, we have some drugs which mimic the reactant that works on an enzyme. And so you can, I don't know, fool the chemical reaction. It works on the drug instead of the reactant. But generally speaking, enzymes are very specific. And for this class, we're going to say that one enzyme works on one specific reactant and then converts that reactant to a product. That's not entirely true, as I stated, that an enzyme may work on something that's very similar to the reactant. And the only examples I can think of are we have some drugs which will take the place of the reactant in the enzyme. The specificity of an enzyme comes to its three-dimensional folding pattern. Specifically, its three-dimensional shape, so that when the enzyme is in its three-dimensional shape, it will be specific to binding to one chemical reaction. The enzyme binds to the reactant, specifically forming a complex, and that happens at the enzyme's active site. Enzymes are not used up or altered in a chemical reaction, so that after the chemical reaction is done, the enzyme is in its same shape, it's in its same form, and it can perform the same chemical reaction countless times. Enzymes do form an enzyme substrate complex, where the enzyme orients itself to the substrate to increase the probability of a chemical reaction occurring, meaning the enzyme will form the enzyme substrate complex that'll put stress on the bonds of the reactant and then lower the activation energy, increasing the likelihood that the reactant will change into the product, meaning the bonds of the reactant will change and become the bonds of the product. And usually that will be breaking a chemical bond or if you're making a larger molecule, making new chemical bonds. Enzymes normally end in the name ase. So like hydrolase is an enzyme. Any word that begins with A, ends with ASE is an enzyme, lyase. We'll talk about ligase, catalase. Those are two that we'll talk about. Not all enzymes end in an ase, particularly enzymes that were named before the naming convention came about. So like renin is an enzyme that curdles milk, especially active in babies. And when the baby drinks the milk, renin is an enzyme that curdles it and then keeps the milk, the liquid milk, in the stomach for a longer period of time, so it is digested longer. My point is renin was named long before a scientist ever came up with, let's call all enzymes ending with ase. Any question about that? Here are some examples of different enzymes. You don't need to know any of these enzymes. We will, however, talk about ligase when we're talking about DNA replication. Enzymes do not work in a vacuum. Sometimes enzymes do need a helper, and that can be either a coenzyme, and that's a biological molecule, usually a small molecule compared to the enzyme, or a cofactor. A cofactor is usually an element like iron or zinc or magnesium. And then that coenzyme, either the coenzyme or the cofactor, binds to the enzyme. You don't need to know the name of the enzyme without the cofactor or coenzyme. You don't need to know the name of it after the, in this case, the coenzyme binds to it. But my point is that enzymes sometimes need a helper. It just depends on the enzyme if it needs a helper. Enzymes do not work in a vacuum. That is, they must work in their correct pH, the correct temperature, their correct salinity. Any question about any of that? There are important coenzymes, so let's talk a little bit more about coenzymes. This graph is showing them. You only need to know the first two. We'll talk about NAD+, which is nicotinamide adenine dinucleotide. It's a coenzyme that is active in aerobic respiration, and we'll talk a lot about aerobic respiration. And that's why you need to know NAD+, because we'll talk about it in aerobic respiration. You should know about FAD also, flavine adenine dinucleotide. We'll talk about this coenzyme also in aerobic respiration. It's based on riboflavin. The other enzymes we're not going to talk about, NADP+ is seen in photosynthetic organisms. Coenzyme A, we're actually going to talk about, but I'm not going to require you to know it as a coenzyme. We'll talk about that in what is that step called? The preparatory step of aerobic respiration and the preparatory step in the Krebs cycle go hand in hand. We call it the second step of aerobic respiration. So this slide is showing you the mechanism of action about enzymes. Let me blow this up. That's sort of blocking it. See if I can get that around. Nope, no better than that. Oh, there it goes. And this is showing you the lock and key model of an enzyme. And that is one specific substrate binds specifically to one enzyme, sort of like one lock fits specifically one key, or better would be I guess one key specifically fits one lock, and why it's called the lock and key model. The substrate or the reactant binds specifically to the active site of the enzyme, forming the enzyme substrate complex that puts stresses on the substrate, which will enhance the substrate to change it into the product. In this case, we're putting stress on the chemical bond right there and breaking it, forming the products. The product then falls out of the enzyme, and that enzyme takes on its original shape and then can bind to another substrate countless times again. Anyways, that's the lock and the key model, where an enzyme specifically binds to one substrate at its active site. This is the hand in the glove model of an enzyme. And what do you notice about the hand in the glove model? What do you notice about this picture here? What's something that's obvious? Is anyone going to comment?
The
Otherwise,
enzyme wraps itself around the substrate.
yes, the enzyme changes its shape when it forms the enzyme substrate complex. And in this case, it's wrapping itself around the substrate. What do you notice about the size of the enzyme compared to the size of the substrate?
The
Nobody's.
enzyme is much larger.
Yeah, the enzyme is much larger than the substrate. That is also the case. These enzymes are biological molecules and they're huge. There's a third thing that you have to really look close, and most of you probably won't get it. I can't seem to blow this up any further. Oh, that's not good. Let me see if I can end this. Oops, didn't mean to do that. And then blow it up here. You might be able to see it now, and that is the substrate changes its shape slightly. It's flatter here than here. Then when it's in the enzyme substrate complex compared to when it's alone and it's just the substrate. And this I like to call the shapeshifter's hand in the glove. So yes, the glove, the enzyme, is changing its shape when the hand is put in it, meaning the glove changes its shape to mold to the shape of the hand. But the shapeshifter's hand is also changing its shape to mold to the shape of the glove. And those of you who have watched Star Trek Deep Space Nine know what I'm talking about when I'm talking about a shapeshifter and how their hand can change to fit the shape of the glove. Any question about that? And this is actually how enzymes work, where both the enzyme is changing its shape and the substrate is changing its shape when they form the enzyme substrate complex, and then that puts stress on the substrate to help change the substrate into the product. You don't need to know, but enzymes work very quickly. The turnover rate is generally 1 to 10,000 molecules per second. And then the enzyme can do this chemical reaction countless times. I don't remember, 10,000 times, 100,000 times, something like that. The enzyme is a biological molecule, so eventually it will degrade and then will no longer function, at least at its optimal rate. I'm not sure if that's just from the enzyme working so many chemical reactions or if it's simply the age of the enzyme, because all chemicals in the cells will eventually degrade. They'll be oxidized by oxygen. Cosmic rays will come from outer space and destroy a molecule they bump into. You don't need to know about that. All right, there are different factors that influence an enzyme's activity. An enzyme can be influenced by temperature, pH, salinity, the presence of heavy metals, the presence of alcohol, and UV radiation. Generally, all of these can cause the protein of an enzyme to denature. The substrate concentration can also influence the enzyme's activity. The substrate, notice, does not cause the enzyme to denature, but when the substrate concentration is very low and the enzyme concentration, we're just going to say, remains constant, you can speed up a chemical reaction by increasing the substrate concentration, because when there's more substrate present, there will be more chance that a substrate will bind to an enzyme, and then the enzyme will change it into the product. You can also influence the enzyme's activity by the presence of poisons like cyanide and fluoride. Poisons usually destroy an enzyme, making it so they cannot work. And I guess if you destroy it, that would be called denaturing it. We'll talk about inhibition. Enzymes can be inhibited. That can be competitive inhibition, where something competes with the substrate for the active site. And there can also be non-competitive inhibition, and that's also called allosteric inhibition, where the inhibitor binds to the enzyme, and the enzyme's in a three-dimensional shape. And when the enzyme now binds to the enzyme, it changes the shape of the enzyme, and that can make the enzyme non-functional. And that's how an allosteric inhibitor inhibits the enzyme. It binds to the enzyme, changes the shape of the enzyme, and now the active site does not bind to the substrate. And then lastly, we'll talk about feedback inhibition and how that can influence an enzyme. Here we're seeing an active functional protein in its three-dimensional shape, and this would be a denatured protein, which you can see is taking on a different three-dimensional shape. And let's say the active site, I don't know, might be right here, binding to the substrate. And I don't even know where that is here, but obviously the active site is not the same. So a denatured protein, if it's totally denatured, it will not work at all. Any question about any of that? There are different factors that can influence an enzyme's activity and influence whether the enzyme will be denatured or not. One is temperature. All organisms, or all enzymes, I should say, have an optimal temperature where they work best. This enzyme shown in this graph is one that works best at 37 degrees Celsius. And where might we find this enzyme? One that works best at 37 degrees. 37 degrees is 98.6 degrees Fahrenheit.
In the human body?
Yes, and most of the human body will be 37 degrees, so this enzyme could be one working in the human body. This probably is not an enzyme working in the body of a cat, because they are warmer than 37 degrees. And anyone who's ever slept with a cat with them in their bed at night, a cold night, will know that cats are pretty warm. By the way, I've never slept with a cat, so just to let you know, I'm not that. I just know the cats are warmer than 37 degrees. I don't know what temperature they are, but they're slightly warmer than 37 degrees. When you move away from 37 degrees, the level of the enzyme activity decreases. When you move far away from the enzyme, the level of the enzyme activity decreases a lot. And what's happening is the protein of the enzyme is beginning to be denatured. And then when you move far away from the optimal temperature, which in this case would be 50 degrees or 10 degrees Celsius, the enzyme has lost all of its activity. It is 100% denatured. And as you know, when you cook an enzyme, for most enzymes that would destroy the enzyme. There are a few exceptions, but generally you boil an enzyme that will kill the enzyme. Any question about any of that? All right. Enzymes are also influenced by pH. They have an optimal pH. This enzyme works optimally around pH 5. Does anybody know where pH 5 would be in the human body? Anyone from A and P?
The stomach?
No, the stomach is more around pH 1.5 to 2 or 2.5, at least when it's acidic and digesting. It would be closer to pH 7 before the stomach produces the acid. So pH 5 is not normally the stomach. Anyone else? Or another guess?
The mouth?
You know, the mouth probably is pH 5. I don't know what it is. I just don't know that one. But the mouth is acidic, I know that. And it's mostly acidic from the bacteria in our mouth, producing acids, digesting food particles, and the mouth is acidic. So that's probably true, as well as the CO2 dissolving in the water of our mouth will make carbonic acid, and that's around pH 5. It might be pH 5.5, but that's close enough to pH 5. And so this enzyme could be in the mouth. What I was trying to say is the skin is around pH 5. So this could be an enzyme in the skin, but like I said, it could be an enzyme in the mouth too, because I know the mouth is going to be around pH 5, if nothing else, because simply CO2 dissolving in the saliva will be making carbonic acid. And so that'll be around pH 5 to 5.5. And then the bacteria digesting food will make acidic products. And so it'll make the mouth acidic. Once again, the enzyme works optimally around its optimal pH, in this case around pH 5. When you move away from the optimum pH, the enzyme will lose activity. You move further away, they'll lose more activity. What's happening is the enzyme is beginning to be denatured. And then you move far away from the optimal pH, the enzyme will be totally denatured and will not function. In this case, pH 0 will be totally denatured, and then pH 10. Another factor that can influence the enzyme activity is the concentration of the substrate. When the concentration of the enzyme is low and the substrate concentration is low, the two aren't often going to bump into each other. So the enzyme cannot easily convert the substrate into the product. If you increase the substrate concentration, you increase the amount of activity of the enzyme, just because the enzyme is more likely to find the substrate and then change it into the product. And that will continue where you increase the concentration, you increase the enzyme's activity to a point, and then the amount of increase starts decreasing. And then we reach this point here where when you increase the substrate's concentration, the amount of enzyme will not increase any further. And the reason why the amount of enzyme activity doesn't increase any further is because all of the enzyme at this point is already bound to the substrate. So if you add more substrate, you do not increase the activity of the enzyme any further because all of the enzyme is already bound. Any question about that? All right. We can inhibit an enzyme by having an inhibitor around. You can have a competitive inhibitor, and this inhibitor competes for the active site with the substrate. If the competitive inhibitor is in the active site, the substrate does not get converted into the product. And like I said, we have some drugs that work as a competitive inhibitor. Any question about any of that? All right. We also have non-competitive inhibitor, and this is also called allosteric inhibitor. This is where the inhibitor binds to the enzyme at a site other than the active site. And that site is called the allosteric site, where the allosteric inhibitor binds to the enzyme. When the allosteric inhibitor binds to the enzyme, it changes the three-dimensional folding pattern of the enzyme so that the active site now is in the wrong shape and conformation. The active site can no longer bind to the substrate. And that is how an allosteric inhibitor works. Any question about any of that? All right, you can have feedback inhibition, and that is when you have a linear sequence of chemical reactions hooked together one after the other, where you start with the substrate and the first enzyme changes that substrate into the first intermediate A, and then intermediate B is changed by another enzyme, intermediate A is changed by another enzyme, enzyme 2, intermediate B, and then intermediate B is changed by another enzyme, enzyme 3, into the end product. Feedback inhibition is when the end product binds to the first enzyme as an allosteric inhibitor, changing the shape of the enzyme so that this enzyme can no longer bind to the substrate. So the end product itself is inactivating the first enzyme. We often find feedback inhibition, this is called feedback inhibition, by the way, that works this way in cells because you're using the end product itself to regulate how much end product is going to be made. When the cell needs more end product, there will be little end product around, so it won't bind to the enzymes and shut them down. And so the enzyme will be active, changing the substrate into intermediate A, which will be changed to intermediate B, which will be made into the end product. As the end product concentration in the cell increases, more and more of the end product will bind to more and more of the enzymes, and you'll start shutting down all of the enzymes. Realize in the cell, there's usually more than one end product molecule, and there's usually more than one enzyme 1 molecule. They're usually numerous numbers. And so when you get lots of end product being made, the cell doesn't really need any more. Lots of end product will mean the end product is most likely going to bind to the enzyme, shutting down enzyme 1. You shut down enzyme 1, you no longer make the end product because intermediate A isn't made. If intermediate A isn't made, intermediate B isn't going to be made, and the end product's not going to be made. The reason why feedback inhibition is often found this way in cells is because it's the most energy efficient. You have the end product controlling how much end product is in the cell. When there's too much end product in the cell, the end product shuts down enzyme 1, shutting down the production of the end product. When there isn't enough end product in the cell, there won't be much end product around to shut down enzyme 1, and then these intermediates will be made so that the end product will be made. And so end product will increase in the cell. And then why we're shutting down enzyme 1 is because it's the most energy efficient. If you shut down enzyme 2, for example, you will make intermediate A, which is not going to be used by the cell because it won't be converted into intermediate B if enzyme 2 is shut down. So if you were to shut down enzyme 2, you're going to waste the energy making intermediate A when you don't need intermediate A. So the feedback inhibition is often seen like this in a cell. Any question about any of this? All right, let's take a step back and state that I've sort of used enzymes and proteins as synonymous, meaning I've said that an enzyme is a protein. That isn't 100% true. Most enzymes are proteins, but not all enzymes are proteins. There are some enzymes which are made of RNA, and these we call ribozymes, and they're simply RNA enzymes. They act like an enzyme, meaning this RNA enzyme cuts and splices a molecule. Usually it's RNA, but it could be another molecule. That RNA has an active site that binds to its substrate, and then the RNA enzymes are not modified or used up in a chemical reaction. So once they perform the chemical reaction, they can bind to their substrate again and perform that same chemical reaction. Any question about RNA enzymes? All right, most enzymes, the vast majority of enzymes in a cell are protein, but there are a few which are RNA enzymes. The only RNA enzyme that we're going to talk about in this class is when we're talking about protein translation, and the ribosome links the amino acids together so that the new amino acid that's brought in to the growing polypeptide chain, which is the growing protein, and it links that amino acid to the growing polypeptide. That actually is done by an RNA enzyme. It's the ribosome that does that, and it's an RNA part of the ribosome. It's not the protein part of the ribosome. And that's the only RNA enzyme that we're going to talk about. Your textbook author does not talk about any RNA enzymes. He does talk about ribozymes, but he doesn't mention any of them. And he doesn't mention when he's talking about the protein translation, how the linking of the newly added amino acid to the growing polypeptide chain, how that happens by an RNA enzyme. Any question about any of that? Tutor, I was trying to remember the name of your textbook author. He doesn't mention that. All right, any questions about enzymes? Any questions at all? If not, I'm going to change gears here and talk about metabolic diversity among organisms. So are there any questions? Everyone's good on enzymes? All right, All organisms, including microbes, require the following. Every living organism requires an energy source, and every living organism requires a carbon source. That's true of us, that's true of microbes. We can classify an organism based on where they get their energy source from. Organisms are classified as a phototroph if they derive their energy from light. On the other hand, organisms can derive their energy from a chemical compound, and these we call chemotrophs. You should note that you can get that chemical compound from an organic chemical compound, such as glucose, or you can get that energy from an inorganic chemical compound, such as sulfate, nitrate, nitrite, hydrogen sulfide gas, iron. Humans only get their energy from an organic chemical compound like glucose, but the chemosynthetic bacteria can get their energy needs from an inorganic compound. Now let me think, is there any other organism besides the chemosynthetic bacteria or organism that can get their energy needs from an inorganic chemical compound? Let me think about that. No animal and plant, obviously. As far as I know, if it's an inorganic chemical compound, it's a chemosynthetic organism. Now, is that true? I think that's true. Organisms can also be classified on where they get their carbon source from. Organisms can be classified as an autotroph if they get their carbon from CO2, either dissolved in the air or dissolved in the water. Green plants are an autotroph as they get their carbon from CO2. Organisms can be classified as a heterotroph if they use an organic source for getting their carbon. Like humans or heterotrophs, we get our carbon source largely from glucose or some other organic molecule that we eat. Okay? Any questions about that? So if ever you are getting a little confused on how to classify an organism, just break it down into what's the energy source. If they get it from a light, it's a phototroph. If they get their energy from a chemical, they're a chemotroph. And then what's their carbon source? If they get their carbon from CO2, they're an autotroph. If they get their carbon from an organic chemical compound, such as glucose, they are a heterotroph. We can then classify an organism based on both the energy and the carbon source for that organism, in which case we have photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs. A chemoheterotroph I'm going to talk about first because we are a chemoheterotroph. Chemoheterotrophs get their energy source from a chemical compound such as glucose, and we get our energy from eating glucose or some other molecule in our food. And we get our carbon source from an organic chemical compound such as glucose or some other chemical in our food. And you'll note that many chemoheterotrophs get both their carbon source and their energy source from the same molecule, which for us is largely glucose, but obviously you could get it from something else you're eating as well. Like we do digest proteins. Oftentimes that is the case with the chemoheterotroph, but it is not always seen that way. Some chemoheterotrophs, for example, can get their energy source from an inorganic chemical compound, such as sulfate, nitrate, or iron. And they get their carbon source from an organic chemical compound like glucose. Like some chemosynthetic bacteria are this way. So all of the fermentative bacteria, meaning the bacteria that ferment things like making of yogurt, animals, all protozoa, all fungi, and many bacteria are chemoheterotrophs. Like E. coli is a chemoheterotroph. Any question about any of that? All right. Another one that's very easy for students to work with are the photoautotrophs. They get their energy source from light. That's why they're a phototroph. And they get their carbon source from CO2. That's why they're an autotroph. Everyone knows green plants are photoautotrophs, but there are green photosynthetic bacteria, such as the cyanobacteria. There are other bacteria which are photoautotrophs, and they may not be green, depending on the pigment of their, what is that called? Their pigment of their light trapping molecule. I forget what that's called, but there is a pigment they have for trapping light. And for green plants, it's chlorophyll. At least that's the main photosynthetic chemical. Some photoautotrophs do not produce oxygen. Obviously, cyanobacteria in green plants do, but there are some bacteria which are anoxygenic, and this would be the green and purple sulfur bacteria. They do not make oxygen. Let's talk about the chemoautotrophs. This will be the next easiest for you guys to understand. The chemoautotrophs get their energy source from a chemical. That's why they're called the chemotroph. And they get their carbon source from CO2, why they're called the autotroph. The chemical that they get their energy source is usually an inorganic chemical compound, like hydrogen sulfide, or iron, or nitrate. And you're actually familiar with the chemoautotroph, you just don't know it. If you ever lift up the back toilet tank, not the toilet bowl, the water above the toilet bowl in the tank, you'll see that brown or yellowish sludge on the sides of the toilet tank. That is actually from a chemoautotroph or autotroph bacteria that is most likely oxidizing iron in the water and then using CO2 for its carbon source. And it gets the energy from stripping electrons from iron. And so you are actually familiar with a chemoautotroph. Any question about that? Now, something I need to point out is that the chemosynthetic bacteria, some of them are chemoautotrophs, and some of the chemosynthetic bacteria are chemoheterotrophs. If the chemosynthetic bacteria obtains its energy source from an inorganic chemical compound and gets its carbon source from CO2, it's a chemoautotroph. If the chemosynthetic bacteria gets their energy source from a chemical compound, inorganic chemical compound, but gets their carbon source from an organic compound such as glucose, they are a chemoheterotroph. So some of the chemosynthetic bacteria are chemoautotrophs, some of them are chemoheterotrophs. And I could throw in a complication here, but we'll wait to talk about the complication. Any question about any of that? All right. The hardest for you guys to discuss and understand are the photoheterotrophs, and that's because you don't know of any photoheterotrophs. A photoheterotroph gets its energy source from a light. That's why it's called phototroph. And they get their carbon source from an organic chemical compound such as glucose. That is why they are a heterotroph. The green and purple non-sulfur bacteria are photoheterotrophs. And this is the only time we're going to talk about the green and purple non-sulfur bacteria. And I'm sure none of you are familiar with the green and purple non-sulfur bacteria. In fact, I'm not really familiar with them either. I've never grown them, never handled them, but they are photoheterotrophs. Any question about this? If not, let me talk about the nutritional classification of organisms. You must know what's in red. First, we have all organisms. We can classify all organisms based upon their energy source. If they get their energy from a chemical compound, they're a chemotroph. If they get their energy source from a light, they are a phototroph. And then we can classify the two trophs on where they get their carbon source from. If they're a phototroph and they get their carbon source from an organic chemical compound, they are a photoheterotroph. If they, on the other hand, they're a phototroph and they get their carbon source from CO2, they are a photoautotroph. And the photoautotrophs you should be familiar with because green plants are photoautotrophs. And let me just remind you that there are green bacteria which are photoautotrophs also. The cyanobacteria is a bacteria that's photoautotroph. On the chemotrophs, which get their energy needs from a chemical compound, we can split them or classify them based on where they get their carbon source from. If they get their carbon source from an organic chemical compound, they are a chemoheterotroph. If, on the other hand, they get their carbon from CO2, they are a chemoautotroph. You don't really need to know that there are photoautotrophs which produce oxygen and photoautotrophs that do not produce oxygen. You don't really need to know that. You should know that the chemosynthetic bacteria can be either a chemoheterotroph or a chemoautotroph. You can split the chemoheterotrophs on where the electron goes to in aerobic respiration, or I should say in respiration. If the electron ends up at oxygen, they are a chemoheterotroph that engages in aerobic respiration. And if the electron does not end up at oxygen, they are a chemoheterotroph that engages in anaerobic respiration. Or in fermentation if the electron goes to an organic chemical compound. We'll talk about that later. All right, any questions about this? Remember, know it's in red. All right. When we're talking about the production of energy, you should realize that nutrient molecules have energy associated with the electrons that form their chemical bonds. And so how we get energy, for example, is eating food, and the nutrients in our food supply our energy needs. And how we get that energy is we break the electrons in the chemical bonds of the food. cells obtain their energy needs from catabolic reactions, and then that energy that's harvested in the catabolic reaction can be stored in the bonds of ATP. So the primary energy storage molecule, and it's called the energy currency of cells, is usually ATP. So when the cell needs energy, it gets that energy usually from ATP. ATP is the energy carrier of cells. ATP has a high energy unstable bonds, and the energy can be quickly released and easily released, which is why cells use ATP to get the energy that they need for some energy needed purpose. Any question about any of that? We'll talk a little bit more about ATP later in this lecture. When cells produce energy, they do so through oxidation reduction reactions. Oxidation is when one molecule or atom loses an electron. So if a molecule or atom loses an electron, we say that molecule is oxidized. Reduction is when the molecule gains an electron. So A loses the electron, it's gained by molecule B. B, we then say, is reduced. Oxidation reduction reactions are always coupled, meaning when one molecule loses the electron, the other molecule gains it. Because oxidation reduction reactions is a mouthful to say, scientists made the term redox to mean an oxidation reduction reaction. So redox is the same thing as oxidation reduction. Here is an example of oxidation where the electron leaves molecule A, so it's oxidized. Molecule B gains the electron, so it's reduced. And here's an actual example. We have copper +1 plus iron, and it's got 3 plus charges. The electron leaves copper, so it becomes copper 2 plus. The electron gains iron. The electron is gained by the iron +3, so it becomes iron +2. So the iron becomes reduced and the copper becomes oxidized. Any question about that? In biological systems, oxidation usually happens where the protein follows the electron. Remember that a proton, a hydrogen ion or a proton is the same thing, and that when you put a hydrogen ion plus an electron together, we get the hydrogen atom. So hydrogen atom is made up of a hydrogen proton, the H plus ion, and the electron with a negative charge. Anyways, because the protons usually follow the electrons, biological oxidations are often called dehydrogenations. Here we're having this molecule lose the electrons, so it becomes oxidized. This molecule gains the electron, so it will become reduced, but more than just the electrons leave this molecule. Both the electrons and the hydrogen ions leave this molecule, and then both the electrons and the hydrogen ion are gained by this molecule. And so we call this dehydrogenation. And this molecule will be oxidized, but it also gains a hydrogen. Any question about any of that? This is actually something we'll look at, and that is one of the molecules starting from glucose will lose its electron, and then that electron will transfer through actually the electron flows directly, it's at least in glycolysis, the electron will flow directly from glucose to NADH, and NADH will then carry the hydrogen and the electron in one place in the cell, in this case, the cytoplasm, to the inner mitochondrial membrane of a eukaryotic cell. You don't need to know that yet, but it will take the electron as well as the hydrogen to the third step of aerobic respiration, and we'll call that the electron transfer chain. And we will talk about all of that later, so you don't have to understand that yet. All right, biological redox reactions, remember redox is oxidation reduction reactions, are used in catabolism to extract energy from nutrient molecules. So when we're engaging in catabolism and we want energy abstracted from the molecule, we will take the energy by using redox reactions. A highly reduced compound is a compound containing many hydrogen atoms. They are degraded, meaning catabolized, to a highly oxidized compounds. For example, glucose, C6H12O6, a molecule which has a lot of hydrogen atoms, can be oxidized to CO2 and water. And this actually happens in aerobic respiration. We'll talk about it. But the point is, the highly reduced compound, the glucose, is degraded to the highly oxidized compounds, the CO2 and water. Along the way, energy is released, and some of that energy is trapped and used to make ATP, so that ATP can be used when the cell needs energy. The highly reduced compounds contain a large amount of energy, and we call this potential energy, meaning glucose contains a lot of energy in it, and that's potential energy. We don't normally see that potential energy come off, but if you were to take a blowtorch to the cup of glucose, I don't know if you can see this, it doesn't have glucose in it, it's got water in it, but if we had a cup of glucose in it, if I were to take a blowtorch to it, I'd have to move back very quickly because the glucose would burn, release so much energy that it would be too hot for me to stay this close to the cup. All right, aerobic respiration is the energy conversion in a cell. It's also called cellular respiration, meaning cellular respiration is used interchangeably with aerobic respiration. I don't like the term cellular respiration, so I only use the term aerobic respiration. And that is, cellular respiration is aerobic respiration in a cell, but a cell can perform anaerobic respiration. Like, for example, most anaerobes do not perform aerobic respiration. They do perform anaerobic respiration, and to me, cellular respiration just is the wrong term to use, but it is used synonymously with aerobic respiration, and I have no idea why that's the case, okay? Anyways, aerobic respiration is shown here where we start with glucose and oxygen. It goes through cellular aerobic respiration, making ATP. Some of the energy in glucose is lost as heat. The glucose and the oxygen are then converted into carbon dioxide and water. Here is the summary reaction for aerobic respiration. Glucose starts it out, C6H12O6. If you don't know what glucose is, you should learn it. Plus 6 oxygen molecules, plus 38 ADP molecules, and 38 molecules of phosphate. That's what PI stands for, inorganic phosphate. Makes 6 CO2 molecules, 6 water molecules, and 38 ATP molecules. Any question about any of that? All right. There's a couple of things here that you'll learn along the way, and that is none of the oxygen will be converted into carbon dioxide. All of that oxygen will be converted into water. All of the carbon dioxide comes from the glucose. We'll talk about that in this lesson. Let's talk about the generation of ATP. Some energy from some redox reactions is used by the cell to make ATP. Animal cells are very poor at making ATP, and they have only two processes for making ATP. That's aerobic respiration and fermentation. All other reactions, meaning catabolic reactions, do not generate ATP. And that's the vast majority of catabolic reactions in our cells. ATP is generated by the phosphorylation of ADP. Phosphorylation is just the addition of an inorganic phosphate group, and this is a phosphate group here, PO4, with 3 negative charges. Gets added onto ADP, shown here. You need energy to make ATP because ATP has more energy than ADP and the phosphate. And then that makes a high energy bond. Please note that ATP has 2 high energy bonds. The third phosphate has a high energy bond, and then the second phosphate has a high energy bond. And that bond is connecting the second phosphate to the first phosphate, or the third phosphate to the second phosphate. Whenever the cell needs energy, it can break this bond, and then that will release the energy the cell needs. And if the cell needs additional energy, it can also break this bond. Any questions about that? All right. Cells can generate ATP in 3 different mechanisms. There's substrate level phosphorylation, where the cell transfers a high energy phosphate from a phosphorylated compound directly to ADP. This actually happens in glycolysis of aerobic respiration, as well as in glycolysis of fermentation, where a molecule containing phosphate gets the phosphate removed from that molecule, and then the phosphate is added directly to ADP to make ATP, substrate level phosphorylation. The second way that cells can generate ATP is by respiration, and this involves the sequential transfer of electrons from an electron donor cell to a final electron acceptor cell. The final electron acceptor is the last molecule to accept the electron from the donor molecule. In aerobic respiration, the donor molecule is glucose. It sends the electron to NADH, which then brings it to, you don't need to know this, but FMN, or is that FADH2? Let me just do that. It brings it to FMN, which brings it to Q, which brings it to cytochrome B, which brings it to cytochrome C, which brings it to, that's C1, to cytochrome C, which brings it to cytochrome A, which brings it to cytochrome A2, which after cytochrome A2, the electron then leaves these molecules and is accepted by oxygen. So in aerobic respiration, oxygen is the final electron acceptor, and oxygen takes the electron as well as hydrogen ions, and then oxygen will be converted into water. Wherever you have this going on, you have a reduction shown this way and an oxidation shown that way. So when the electron is given by NADH to FMN, FMN is reduced, and then FMN will give the electron to Q, so FMN is now oxidized and Q is reduced. That's how you read that. Any questions about any of that? If not, let's talk about the third way that cells can generate ATP. This happens by photophosphorylation. Photophosphorylation only happens in photosynthetic cells where light energy releases electrons from chlorophyll. The light energy is then converted to chemical energy via an electron transport chain, and we'll talk briefly about that at the end of this lesson. ATP and NADPH are generated. Oftentimes, the ATP and the NADPH are then used to make sugar. So we make ATP, but then we degrade the ATP into ADP to make the sugar. As I mentioned, photophosphorylation only happens in photosynthetic organisms, which we're seeing a bunch of photosynthetic organisms here. That's why the boys are so muddy. That's a joke. You can see some photosynthetic organisms in the background, though. Lots of them, actually. All right, any questions about that? How cells make ATP? 3 ways. We will talk about photosynthesis at the end of this lesson, and then a large part of this lesson will be talking about aerobic respiration. You'll note I didn't say aerobic respiration here, I said respiration. That means both aerobic respiration and anaerobic respiration. All right, let's talk about energy production. Energy is released from molecules in controlled ways and stored by a series of reactions, meaning the energy is not released in a single energy burst. For example, here, we're digesting glucose. Glucose gives its electrons to NADH, NADH to FMN, and then the electron transfers to each of these molecules until it eventually reaches oxygen. And at each step along the way, the electron loses some of its energy so that the energy is lost in numerous steps along the way. It's not lost in one burst of energy. And that's why the cell can burn glucose at a temperature relatively around 37 degrees, if we're talking about your cells. And you're releasing the energy from glucose slowly. If we were to release the energy all at once, you can do that by simply taking a blowtorch to it, and the temperature of that glucose burning would be around 1,000 degrees, which would, of course, fry your cells. Electrons from the donor molecules are passed from one compound to another through a series of redox reactions. And there we're seeing the redox reactions. Most organisms can use carbohydrate catabolism to obtain their cellular energy needs. Now, not all organisms do this. For example, green plants, when light is out, do not get their energy needs from carbohydrate metabolism. But all animals do, and many bacteria do it as well. So we're going to talk about carbohydrate catabolism. The most common energy source is glucose, although cells may use lipids and proteins to get their energy needs, meaning we can digest lipids and proteins. To produce energy from glucose, microorganisms use two general processes in carbohydrate catabolism to generate their energy needs. They can do it by respiration, and there's two forms, aerobic respiration and anaerobic respiration, and they can do it by fermentation. Let's watch the little video on aerobic respiration.
Let's say I'm working on a research paper about the future of renewable energy, and I come across a study with findings I want to include in my paper.
All cells need energy to stay alive. Usually your cells get their energy through the process called aerobic respiration. The word aerobic refers to oxygen. In aerobic respiration, oxygen interacts chemically with a molecule of glucose. The glucose is broken down into carbon dioxide and water. This process releases energy, which the cell stores in the chemical bonds of ATP. The first stage of aerobic respiration, glycolysis, occurs in the cytosol, the jelly-like part of the cytoplasm. Glycolysis breaks each molecule of glucose into two molecules of pyruvate, energizes two electron carrier molecules, and produces two molecules of ATP. The next two stages of aerobic respiration are carried out in the mitochondria. Pyruvate enters a mitochondrion. Once inside, it is modified and enters the Krebs cycle. The reactions of the Krebs cycle break the pyruvate molecules down to carbon dioxide, create two more molecules of ATP, and energize more electron carrier molecules. The final stage of aerobic respiration is electron transport. The electron carrier molecules that were energized in the earlier stages deliver electrons and hydrogen ions to the electron transport chain. As the electrons are passed down the chain, energy is released and used to make ATP. This final stage produces as many as 32 molecules of ATP from each molecule of glucose.
All right, any questions? If not, I'm going to stop here, and I'll see some of you at 7 in the lab.
Thank you.
Thank
You're
you.
welcome. You're welcome.